All right, welcome everyone. Let's talk about this new variant. It is all over the news. Every single news outlet has multiple news stories about it. I've been looking around for actual scientific papers about this. There are none. This is the B.1.1.529, other now recently called the Omicron variant. The World Health Organization has called this a variant of concern. They met on November 26th, 2021, and that is today, and they called it a variant of concern. A variant of concern is what they have to do. It has to have increase in transmissibility or detrimental change in epidemiology, or increase in virulence. So that means it's more, so it either transmits easier, or it's more virulent, or it decrease in effectiveness in public health and social measures of available diagnostics, vaccines, or therapeutics, which means it either cannot be tested for, so it doesn't show up on tests, or two, the vaccines don't work for it, or three, the therapeutics that we have don't work for it, or all three of these things. So this Omicron variant has features that make people think that it might have all three. It might be it seems to be more transmissible. We don't know about virulence yet, but it's likely to evade vaccines to some extent, and it's likely to evade therapeutics to some extent. We don't know. It's all kind of conjecture at this point. People are worried, and I'll show you why they're worried, but we don't have strong data yet, and that's forthcoming. So, but there's a lot of there's a lot of papers, a lot of articles in like Fox News, CNN, Wall Street Journal, New York Times, The Guardian, BBC. It's all over the place. So they're talking about it. After looking at all this, the only actual primary data that I found is this, and I will link this below. It comes from South Africa. This is their network for genomic surveillance in South Africa. So South Africa is actually doing a pretty good job of doing genomic sequencing of the viruses that they get in the public, and so they can find new variants and keep track of how variants are moving through. So this is the summary. New variant was detected in South Africa, lineage B.1.1.529, with a high number of mutations, and which are predicted. So these mutations, based on laboratory data and previous variants, they think that it's going to increase transmissibility and increase immune evasion. This was found in 77 samples from this area of South Africa called Gauteng, and then 4 in Botswana and 1 from Hong Kong. And that one from Hong Kong was a traveler from South Africa. Okay, so here's the timeline. Tuesday, November 23rd, there was a new variant that was discovered, and then they had a meeting to go over this variant because it had so many new mutations. And this is the thing that's concerning to them, is because if you look at the reproduction number, that is how quickly the virus is spreading. It was 1.4. And then in Gauteng, which is the region in South Africa where this variant is prevalent, it's 2. So it's significantly more transmissible than the Delta variant, which has been going around. So if you look at, this is their mutational profile. There's over 30 mutations in the spike protein, as well as several mutations in the nucleocapsid and other proteins. But the spike protein is the one that we are kind of most interested in, because that's the one that we target with our drugs and with our vaccine, and it is the mechanism by which the virus enters our cells. So mutations in the spike protein change how well it infects people. All right, so they go over which of these mutations may cause problems. So they said there's multiple receptor binding domain and N-terminal domain mutations with resistance to neutralizing antibodies. So there's lots of mutations that are in the area that neutralizing antibodies that people make after they've been naturally infected, as well as from vaccines, as well as the therapeutic monoclonal antibodies like the Regeneron monoclonal antibody. They all target that area. So if there's mutations in that area, those antibodies may not work. There are a couple of mutations here noted next to the furin cleavage site that laboratory tests show may have enhanced transmissibility. That means they're more efficient at cell entry. So if you get exposed to this virus, it's more likely the virus is going to infect you. Then there's a couple of mutations that are similar to previous variants that may evade innate immunity, so T cell mediated immunity. And then there's a couple of mutations in the nucleocapsid that are associated with infected infectivity. This is the other thing that's making them very concerned. You look at this, if you look at this timeline in South Africa of what the dominant strain was, the dominant strain was the beta, which is the South African variant. And then that got overtaken by Delta. And then if you look at this, the blue, the B.1.1.529, or the Omicron variant, is now rapidly overtaking Delta. So it's going to become, at least in that region, it's becoming the dominant virus. So it's outcompeting the Delta. The Delta outcompeted the Beta, and now this one is outcompeting the Delta. So that is concerning. All right, so that's the end of that one. There is a nice color-coded thing that Dr. Barrett, Jeffrey Barrett, he is a scientist on Twitter, so he kind of color-coded this for us, so it's a little easier to figure out. So basically what this tells us is the red areas are the areas that we've seen previous mutations on. The green is like things that we don't care about because everyone has it. It's a very common mutation that's been had for a long time. And then the purple are the areas that laboratory tests suggest that they may be important. So if you look at this, these are things in the alpha variant, the delta variant. These are probably transmissibility things. The beta variant, you remember the beta variant, which is a South African variant, was called the South African variant, was the variant, if you've been following this channel, that's the one that I was worried about that had a lot of immune escape, and I thought that would become the dominant variant, but Delta was overwhelming it with more viral load numbers. So Delta actually became the dominant one over Beta, but Beta had all these immune escape mutations. So here's a beta mutation, and then some gamma and alpha mutations here. And then these purple ones, see, these are near the, this is monoclonal antibody binding. So these are the binding sites for a lot of the monoclonal antibodies, as well as the neutralizing antibodies that you get from vaccination or from natural immunity. So they will bind in this region. So this makes people think that this thing could evade vaccines, natural immunity, and monoclonal antibodies. We don't know that. This is all theoretical at this point. People are testing them right now, and they will see if it actually is significant. This may turn out to be a lot of nothing, or it may turn out to be very significant. We don't know. And then these purple ones are predicted to increase the binding site. So I just wanted to show this, like Moderna and all the other companies are looking into this, and I'm sure like Regeneron and all the monoclonal antibodies are going to be testing it. Once they get this sequence, they're going to put it into a plasmid. They're going to make the new spike protein that this variant has, and they're going to test it against the neutralizing antibodies that they have in their freezers with these vaccines, as well as against these monoclonal antibody therapies. So we are going to get that information pretty soon, I think. And Moderna, what they're doing, and I'm sure Pfizer and the other companies are doing the same thing, they're testing a multivalent booster, which they've been working on a booster or a specific one for the beta variant, which has a lot of the same mutations as this variant. It kind of is almost like this Omicron variant is like Delta plus Beta. That's not the lineage, that's not how it came to be, but it shares a lot of similar mutations between the two. So it's kind of like you took the worst part of Beta and the worst part of Delta and put them together. So that's what they're doing. They're testing a multivalent booster, and they are trying to make a Omicron specific booster. Since this is just the RNA vaccine, they can iterate and make new vaccines based on new sequences very easily. So anyways, I just want to give you guys a heads up on what this is. The only primary data source is this, which I will link, and you're welcome to look at it. And there's basically a dearth of information from actual scientific sources that have tested it. They are being done at the moment. This sequence is being put into plasmids if it has not been already. I'm sure they're putting into plasmids, expressing it, and we will see information about how well neutralizing antibodies work against this base, both from natural immunity and from vaccine-mediated immunity. And then we will also see if it evades the monoclonal antibody therapies. And then as this becomes more prevalent, we will see if it does cause more severe disease or not. Those are all kind of questions we don't know. What we do know is that at least in Guatang, it is outcompeting Delta, and it does have a high R number of 2. So it is transmissible, and it is outcompeting the dominant variant at this time. So it does seem significant in that respect. We don't know about immune escape or therapeutic escape yet. That information is pending. As soon as I hear about it, I will kind of make an update video about it, because I'm interested myself. And thanks for watching. Any questions, put them below. But right now, I probably can't answer them. Probably no one can answer them, because we just don't know yet.